You speak of the case in Australia. Yeah. Well, in Australia there was a man who it's claimed infected 3 women, and I think 1 woman died. In the defense of the man, I don't know the details, if he did it deliberate, if he knew. I don't want to get into that and what the punishment should be. But the question is his defense was an absurdity, but that the virus didn't exist.
The biggest problem with the HIV theory of AIDS is HIV.
There is a group of AIDS denialists that say that HIV does not exist and has never been isolated, which is as bizarre as it gets.
You and your colleagues not only state that HIV does not cause AIDS, but you take an even greater leap and say HIV does not exist. Is that correct?
Oh, it is partly correct. We do not say that HIV doesn't exist. It may exist, but the presently available data does not prove its existence.
But how can you say that when world-renowned scientists like Dr. Gallo and Dr. Fauci say HIV does exist? Are you telling me and the world that they're all wrong?
No, what we're saying is there is the evidence there, the data in the scientific literature, which is published. Scientists interpret data differently, and we interpret the evidence one way, and they interpret it in a different way. So in our view, the evidence does not prove the existence of HIV.
We've all seen pictures. We've seen electron micrographs of HIV. How can you say something that we see isn't there?
You didn't see electron micrograph of HIV. What we see is electron micrograph of particles which look like retroviruses. But it's one thing to look like and another thing is to be a virus.
The one thing I don't understand is how can you question the existence of a virus when there was an international lawsuit against the United States government and Robert Gallo for stealing the French virus? I mean, it seems to me there must be a virus there if somebody stole it.
That's the problem. Under no circumstances Robert Gallo could have stolen Montanier's virus, even if there was such a virus. Because what Montanier sent to Gallo was supernatant from his cultures. And in the supernatant, the viruses don't last for too long. And in fact, the particles have to have knobs on their, or spikes on their surface to be infectious. And these knobs are lost. And in fact, nobody has proven that they exist. But even if they exist on HIV, or the particles, they are lost very rapidly. So it is impossible for Montanier's, for Gallo to have stolen a virus from Montanier.
So can you prove to me that HIV doesn't exist?
I can show you what the evidence shows, what Montanier, for example, presented, because everybody accepts that Montanier is the discoverer of HIV. And I can show you the evidence which Montanier presented and claims to have proven the existence of HIV. And I will explain to you why Montanier's evidence does not prove the existence of HIV.
What do we mean by virus isolation or virus purification? These are jargon words in virology, and they're not very precise. They mean different things to different people.
Now, Dr. Gallo and Dr. Fauci talked a lot about isolation and purification. Can you tell me what the differences between the two?
Isolation, what was it?
Isolation and purification.
Of the virus?
Yes.
Well, you isolate a virus by finding the virus which causes a disease. You purify a virus by making a lot of, just by purifying it so you get a pure virus.
Okay.
I don't understand what the issue.
They interchanged the two, and I wasn't sure
I see.
If it was the same thing or if it was two totally different.
No, it depends on how they use it.
Okay. Can you explain the process of HIV isolation?
Well, didn't Dr. Gallo do that? I mean, he actually isolated it. So,
Yeah.
I mean, why should I do all of this? This is all textbook stuff you're asking me.
I'm not quite sure what's behind your question about isolation.
I don't want to be your textbook. You know? I got other things to do.
Isolation is essentially getting the virus from the patient and being able to transmit this virus to another cell to reproduce the infection and to have a continual supply of the virus, and that's called an isolation. Purification is just obtaining the virus free of cellular contaminants or other contaminations, but it doesn't mean necessarily that the virus is infectious.
Virus isolation I would take to mean that you take some infected material, like a blood sample from someone who you think or believe may have HIV infection, may already have symptoms of AIDS, and you try and grow the virus from that blood sample. So you would put the blood sample into culture, you would stimulate the lymphocytes to proliferate with various growth factors or cytokines, and you would harvest the virus from the culture medium that you're growing it in. You would spin out the cells, separate the cells from the supernatant, and you would look for the virus in the culture medium. That needn't be a pure medium, because you can use markers of the virus that tell you it's there, such as the reverse transcriptase enzyme. So you can see evidence of the virus without actually purifying particles. In that very first paper from the French group, published in May 1983, there were two things that appeared to class it amongst the retrovirus family. One was reverse transcriptase activity, and one was actually looking at virus particles with the electron microscope.
The title of the 1983 paper by Montagnier and his colleague is Isolation of a T lymphotropic retrovirus from a patient at risk of acquired immune deficiency syndrome, AIDS. Now, the word isolation means to obtain something or to place something apart or to obtain a substance separate from everything else. Apparently, Montagnier by isolation did not mean this, but something totally different. And I'll explain to what he meant by isolation. He did three main experiments. The first experiment, he took lymphocytes from a patient, now is known as BRU, and he cultured them with many substances, including PHA and other growth factors.
What are growth factors?
They are substances which make cell to multiply.
Okay.
Right? Or to live, and to stay alive in the culture. And after 15 days, he discovered their reverse transcriptase activity. And they claimed that this proved the existence of a retrovirus in their patient, in their patient's cells.
So every 2 days we were taking the supernatant, looking for the presence of the enzymes that transform RNA to DNA, the reverse transcriptase, because we know that this enzyme is associated to virus particles. So we were testing for this enzyme activity in the supernatant.
What's so special about reverse transcription?
Anytime you're searching for a new agent, you want to have some simple measurement of the presence of that agent. In times past, you would put it on cell culture in the laboratory, and suddenly these beautiful cells will all start turning into dead cells. You say, oh, something's there. And then you put that into an electron microscope and look and you can find it. All those are rather difficult things to use. If there's something that the virus produces in this culture, you don't necessarily have to see all the dead stuff. You just can take off some of the liquid it's growing in and test it. And one thing you can test for a retrovirus is reverse transcriptase. And it just happens to be that's a laboratory test available for it, so you just take a little bit off, put it into a chemical assay, and you can do it very, very simply. So it's a matter of something that you can put a lot of specimens through and something that's simple to do, so you can really get maybe not a direct picture of the virus, you can't see it, but you can get evidence that it is there, like fingerprints.
Then he took the lymphocytes, which originated from bruised lymph nodes. He took these lymphocytes and cultured them with the lymphocytes of a healthy blood donor. And there, again, he detected reverse transcriptase activity. So Montagnier found reverse transcriptase activity, and according to him, this proved that the retrovirus was there. But the only way to say that the existence of reverse transcriptase or the detection of reverse transcriptase activity proves the retrovirus was there is only if reverse transcription was specific to retroviruses, which is not the case. In fact, today nearly everybody accepts that reverse transcription or reverse transcriptase activity is non-specific to retroviruses. In fact, at present, everybody accepts that reverse transcription is present in all normal cells. In fact, as far back as at the beginning of the 1970s, Gallo himself have shown that normal cells, when put in culture and they're stimulated or they're cultured with PHA, they will start having reverse transcriptase activity. Montagnier put the bruised cells in a culture. Into culture, he added different growth factors, including PHA. And after 15 days, he detected reverse transcriptase activity. However, PHA by itself in normal cell, and this was known by Barré-Sinoussi, the principal author, and Gallo proved it at the beginning of the 1970s, that PHA itself causes reverse transcription in normal cells. So he put something in the cell which was causing reverse transcription, and yet he said that this proves the existence of a retrovirus there.
So you're saying that what they found might just be the actual substance they put in the culture and not a virus?
Definitely. That is their evidence. And
why would they do that?
Barré-Sinoussi knew that in 1973. Gallo proved it at the beginning. In 1972, I think, was his paper. I don't know. Now, everybody accepts that reverse transcriptase activity is a characteristic of retroviruses, but it's not specific to retroviruses.
But in all my interviews with scientists, they all say that reverse transcription is unique to retroviruses, and that's how they knew that there was a virus in their culture.
Once you have produced, you produce something in the extracellular medium, you can do actually several things. One thing is that if we expect or suspect it's a retrovirus, like HTLV1, the leukemia virus, what we can do is look for what we call reverse transcriptase activity, the enzyme, which is unique to these viruses.
Yeah, the scientific procedure was multifaceted. I had a good-sized lab, and so we had divided people up into different primary goals, and would meet daily. And the first goal was, if a patient has fully developed AIDS, forget risk groups at the time. We didn't want to get into the risk group. They had AIDS, documented AIDS. Could we take their blood, get their T cells, like we would do with any like the leukemias, like we did with the leukemias, the same protocol as we did with leukemia. Take those T cells, clean them up from other cells, grow them with interleukin-2, our old friend, and see if we could find any evidence of release of virus particles, and if we were right that they would be retroviruses. And how to do that was with a surrogate marker. You don't use electron microscopy in those days, except maybe 1 or 2 pictures just to confirm or to see the structure of this particular retrovirus. The assay was reverse transcriptase.
Well, I don't know whom you interviewed, but if you don't believe me, go and ask Baltimore or Vermouth, and I'm sure they will confirm that reverse transcriptase activity is a characteristic of retroviruses, but it's not specific to them.
What do you mean when you say reverse transcription is a characteristic of retroviruses, but not specific?
Oh, let me give you an example. Hair is a characteristic of humans. You know, black, white, or yellow, we all have hair, but it's not specific because there are many animals, for example, cats, dogs, which also have hair. And finding a hair in a room, it doesn't mean that a human being was there, or a cat or a dog.
Are retroviruses the only ones that can reverse transcribe?
No. There are other forms of reverse transcription that are used in various ways inside the cell. For instance, the ends of chromosomes are made by a reverse transcription process. That's how they're maintained stable. There is reverse transcription in the inheritance of all of our cells, which comes about from endogenous genetic elements in the cells, or in the cells of our ancestors. Because once that information gets into our cells, into our genomes, it stays there forever. So it could be that we've inherited information from monkeys or from other animals that are in our lineage. And so, no, reverse transcription is actually very widespread. Something like 50% of the DNA in our cells comes about by reverse transcription.
Oh, that much?
Yes.
Oh, wow. Okay. I thought it was 1 to 2%. Going back to, I thought it was 1.5 to 2%. The 50% the retroviral DNA, or retroviral genes in our DNA, what biological function do they serve?
Now, we got to make a distinction. When I said 50%, I'm saying 50% of the DNA came about by reverse transcription. But it's not all retroviruses. Lots of it is just repeated elements that are there as what we generally consider to be parasitic DNA. DNA which is, as other people call it, selfish DNA. DNA which is in there because it's able to copy itself and reintegrate itself in other places. And this is something that's going on all the time. And it builds up.
Why do you think they used reverse transcription to prove the existence of a virus if they knew that it wasn't specific to viruses?
I don't know. It's as simple as this. I don't know. In fact, they went one step further. In the second experiment, they have taken the cells from their patient and mixed them with cells from a normal blood donor. And in that culture, again, they put PHA and other growth factors, substances which make the cell to survive and grow. And again, they found reverse transcriptase activity. And this time, they claim that the finding of reverse transcriptase activity in the second culture proves that the virus was transmitted from the patient's cells to the normal donor cells. And in fact, they say that this proves.
They must have used some other criteria to state that they had a new virus.
There was no other criteria. In the first, they had a third experiment, but in the first and second experiment, that's how they are described in their paper. It was the first experiment, reverse transcriptase activity, the second experiment, reverse transcriptase activity. They are the only evidence they have published.
So in the
from the first and the second experiment.
So in both experiments, they fed the culture substances which artificially cause reverse transcription?
Yes. Yes, they fed them. Just one of them, PHA, by itself, it will cause reverse transcription in normal cells, in non-infected cells.
What are your problems with the third experiment?
Well, the problems, first of all, again, they took the supernatant, the fluid from their second experiment, and put it in a culture which contained umbilical cord lymphocytes. And in that culture, they also found reverse transcriptase activity. Again, the reverse transcriptase activity, they already use there this PHA and other substances which can cause reverse transcription in any culture, if the conditions are right. Then we have the virus particles. They have published electron micrographs showing virus-like particles budding from the cells and released in the supernatant. But just seeing virus-like particles is no proof that they are viruses, because you can have virus-like particles and they're not viruses. In fact, the third problem with the virus, with the particle, is that they use umbilical cord lymphocytes, which originate from placenta. In placenta, it was known in the 70s that they release virus-like particle and they have reverse transcriptase activity. So find them in this culture is no proof that they originated from Montanier's patient.
In Montanier's paper, they publish electron micrographs of HIV.
No.
We see pictures of the virus.
We see pictures. These particles were in, as I said, in umbilical cord lymphocytes. And umbilical cord lymphocytes originate from placenta. And there was already a lot of evidence by this time that placenta can release virus-like particles. What's more is Montagnier, when he described the particles he has seen, he said these particles are typical type C particles. Retroviruses are classified in different categories, one of which is type C particle. And they said that the virus they have seen is a typical type C particle. Typical type C particles have been many times published in the 1970s from normal placenta. In fact, most of the placentas was shown to have reverse transcriptase activity and typical type C particles. So finding these particles there, even if they were virus, they could have been just originating from the normal placenta. So finding the particles and finding reverse transcriptase activity in the umbilical cord lymphocyte culture does not prove that the patient was infected with a virus.
The two main things that told you had a retrovirus was the reverse transcriptase activity and the fact that the cells died.
And then we immediately call our guy who was responsible for electron microscopy and said, please, could you look under the microscope whether you can see virus particle and if it resembled to a retrovirus.
Okay.
Okay.
And what did you see? What did you guys see when you looked under the electron microscope?
And after quite, it was very difficult because it was only few cells infected, so it was a very difficult task for him to find a cells that was just producing this particle, but finally he found it. And he found one lymphocyte with a budding particle, typical of retrovirus, and very close from these cells, one complete mature particle that resembled to a retrovirus.
In that paper, he had only one electron micrograph, and the virus could be identified as sort of a retrovirus, but it could have also been an arenavirus. Now these are specifics that are only important for experts. But when we saw that photo, we said suggestive, but not convincing.
I've seen these publications, stamp-size images. It's a nuisance. it's a nuisance. You do not really see much. And I do not know how good electron microscopy was really done, but they probably should have done a bit more, and then they would have been very safe. And electron microscopy is an art and a science. Art and science, and nobody is able to judge what the electron microscopist is really doing. You have to have experts discussing these things. Sometimes people are far from the microscope. They do sometimes, and that's a
way, if you want to say that these proteins are from HIV, you must have them coming from HIV. But the particles are too small, so the best next thing is to purify. And this is not me who says it. I mean, this is all the trophorologists, including Montagnier, including Charmaine, including Barré-Sinoussi. That is, this is so simple. So you must have purification. That is the only way. You see, this is what HIV. Retroviruses have the matter of 100 to 120 nanometers. They have a cone-shaped core. They have the lateral bodies, and they have knobs.
What are the knobs?
When we see HIV being diagrammatically represented, you see there's some spikes coming out from the particle. And these knobs are extremely important for effectivity. Again, according to all the HIV experts, the knobs are crucial. They are essential for infection. No knobs, no infection. And there is no infection, the particles cannot be viruses.
So a complex process of an interaction of the outer protein of the virus, called the envelope, with molecules on the surface of the cell are essential for a virus to enter the cell. And when we speak of entry, we essentially mean the gut. It's genetic information in the form of RNA and certain other proteins.
Today, nobody has proven the existence of knobs in the cell-free particles.
Obviously, when we first were working on the cause of AIDS, we had to be very broadly have our ability to include a whole variety of different potential organisms that could cause it. What was helpful in the limiting of our search were the cases in hemophiliacs where the material that's used to treat hemophilia comes from blood or plasma, a subcomponent of blood that is sold or donated into the system, and then a purification process is used to take that large volume of plasma and specifically pull out the anti-hemophiliac factors that actually can treat the hemophiliacs. Now, in that process, the one thing you have to realize is that material is given intravenously to the hemophiliacs to treat them, and you have to have it very clean. You don't want to have any infectious agents in that if you can avoid it. And one of the easiest ways to do any of preparation of a drug is to filter it. Now we have these wonderful modern filters where you can work on your drug, and then the last step before you put it in the bottle, pass it through a filter that filters out all infectious agents. Now I say all, meaning that the filter can only filter out those that the filter can catch, which is the bigger agents. That bacteria and parasites are much bigger than viruses. And those filters indeed will filter out all the bacteria and make it, quote, sterile. That is, you cannot grow bacteria from it. But yet we knew that after this process that they were still getting this disease, which could have been caused by a virus, a bacteria, or parasitic disease, or whatever. But since all of the larger bacteria and parasites are eliminated in these filters, then you can assume that the disease that you're looking at is caused by a virus.
And does it also filter out the infected cells? So is it just pure free-floating HIV virus?
The factor VIII material is cell-free, so it's only liquid material, and that liquid material could contain many infectious agents from the donors of all these. Literally, there's hundreds of donors in each of these batches, and when you filter it out, you filter out all the bigger organisms, that is, bacteria and parasites. You're only left with viruses that go through the filter.
I reckoned that a virus that would survive the purification of clotting factors from human blood was more likely to be a virus without a cell membrane, a small virus like a parvovirus, and I would not have put a retrovirus at the top of the list. And yet, when HIV was discovered, it was indeed a retrovirus. I think my scientific reasoning was perfectly correct, but
Factor process?
well, I didn't realize how unharsh the purification of clotting factors is. We were talking about purification. Those clotting factors are anything but pure, and they were tainted with this virus. They were tainted with hepatitis B virus, with hepatitis C virus, which also was not discovered until long after HIV was discovered. So HIV was just one of several viruses that was passed in clotting factors.
Here it is a paper which was published by several authors, with a co-author Geldenblum, and they say they have found that on the average, immediately after release, after the knobs are released, as particles into the fluid, there are 0.5 knobs per particle. And they said even these 0.5 knobs, they may be false positive. That is, there are no knobs at all.
The knobs here look like little knobs everywhere.
Yeah, okay. There's a chance that you have some dirt, of course, in the cell culture, and be knobs. Here's the same. That's my interpretation. So my interpretation. But here are the knobs. Here might be there might be some dirt. Here, sure, that is dirt from preparation.
Mm-hmm. But don't these look like knobs too, coming off of this big one?
Yeah, you know, there are glycoproteins also on the cell.
Oh, okay.
Yeah. Now, we have to differentiate between them. If we would like to go for differentiation, we have to use immunoelectron microscopy to make a tag. It's either the glycoprotein or it's something different.
In a paper published in 2003, the author state the clothes of GP180, that is the knobs, do not form spikes on the surface of HIV, as is commonly described in the literature. We suggest the spike knob observed by negative staining electron microscopy may be an artifact of the penetration of heavy metal stains between the envelope proteins.
What exactly does that mean?
That means that despite some people claims that there are knobs on the HIV particles, these authors say that the particles have no knobs. In plain language. This is a paper published 2006 in Nature. Here it is SIV, simian immunodeficiency virus. And in these particles you can see many knobs. But when you come to the electron HIV particles, there are hardly any knobs. In fact, but you can see the same thing down there. And you don't know if there are knobs or there is some artifact there. In fact, the authors call them putative knobs. Or putative means supposedly HIV knobs. Mm-hmm. So the authors do not have, even today, proof that the HIV particles have knobs which are crucial for infection. Even if we admit, even we accept that there are knobs, there are 0.5 knobs immediately per particle on the average, immediately after they are released, they are lost. Even this small number is lost very rapidly, according to Geldenblum. And these knobs being lost so rapidly, And factor VIII preparation taking a long time from the time the blood is taken to the time factor VIII is prepared is a long time. And even after it is prepared, may stay on the bench for months before it is used. So it is impossible for hemophiliacs, the factor VIII, to contain an infectious HIV particle, even if HIV exists.
I wanted to ask you about hemophiliacs. Because they had cell-free plasma, it was just the virus. But the virus sheds its membranes within 24 hours. So one thing we couldn't get is how was it able to actually infect the T cells?
Yes, it's a question, but we have to know that all the fraction of the blood could be infectious. And there is some virus bound to red blood cells which could be released also in the plasma after treatment or incubations. Some virus is bound to red cells also as well. So perhaps there are more virus when you process the blood, more virus could come in the plasma, okay? And these virus could be protected by the plasma proteins from denaturation. This is one thing, one possibility. The other is the quality of the host. Hemophiliacs are fragile. Being transfused many times, their immune system also is depressed before they get infected with HIV. So they are prone to HIV infection because of their immune system weakness. Okay? So combining this with the fact that the virus could exist also in forms which are metastatic, we think for the usual particles,
label them HIV because the reviewer asked them. So the question is why they didn't label the proteins higher than 41. Well, there are some good reasons for it. First of all, many HIV experts, maybe not all of them, but many HIV experts accept that the protein of 120 and 160 are polymers of P41. That is, they are 41 joined together. 341 are joined together and are made 120. 441 are joined together and are make 160. They admit all the proteins above 24, there is evidence that the protein above 24,000 molecular weight are cellular proteins. And they also know that the proteins, well, is molecular weight lower than the P70 and P6, P7, they are subunits of proteins with a high molecular weight. So now we are left with one protein, P24. We come back. Now, what is 24? Well, to answer that question, we have to go to an interview which Montagnier gave to the French investigative journalist Jamel Tahi. Jamel Tahi asked Montagnier why they didn't publish any electron micrographs of the material which they said represented purified virus. And Montagnier responded, we found some particles, but they did not have the morphology typical of retroviruses. Jamel Tahi insisted to find how it was possible that they did not publish when they claimed that they have purified it, and then in the purified virus they did not have typical, even retrovirus purified. Montagnier repeated, I repeat, we did not purify it. Then he was asked if Gallo purified it, and Montagnier responded, I do not know if Gallo, really purified. I don't believe so. Jamel Tahi asked Montagnier, do any picture from the for purification exist? Montagnier responded, yes, of course. Have they been published? Montagnier, I could not tell you. We have some somewhere, but it's not of interest, not of any interest. For us, it's of very great interest. It's crucial. If you don't have them, then there's no way you can prove the existence of HIV. And then we ask God,
like that, but it was necessary also for us to control, because this house in 85 already established ELISA antigen material for testing people. There was nothing commercially available, but we had to purify. We had to look at the material that was used for the ELISA. 80% dirt. Okay.
Okay.
That's the truth.
Again, Jabel Tahi interviewed Charles Dauger, which was the electron microscopist at the Pasteur Institute. And in his response, he said, what in the purified virus, they had only cellular debris. They never had virus. Which means that the P24 protein originated from material which contained not even retrovirus particles, much less purified virus.
So in 2001, the EM specialist from Pasteur admitted they never saw anything.
Yes. Yes, he admitted that they never saw any virus-like particles, much less purified virus. In fact, what they had there was cellular debris all day long.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus, you know.
The P24 originated from a material which did not even have virus-like particles, much less purified virus.
So it's cellular debris?
Yes, had only cellular debris, which means you cannot have a better evidence that P24 is a cellular protein, cannot be HIV. If it originated from a material that contains only cellular fragments, it has to be a cellular protein. Now, even if we assume that P24 is an HIV protein and that Montagnier discovered the first one-protein virus, this creates even a bigger problem. This is because retroviruses have an enzyme called reverse transcriptase. Called retroviruses. Their name derives from this protein. The virus cannot be a retrovirus. That would be like having an object which does not have any wings, and yet you still call it. Experiments, Montagnier found reverse transcriptase activity, and on the basis it did not have any, they call this RNA HIV RNA. It's even worse. Gallo knew as far back as 1972 that adenine-rich RNA is not specific to retroviruses. This type of RNA can be found in any cells which are synthesizing proteins. In any case, the existence of HIV and even its causative role in AIDS were both accepted well before any genomic studies were published. Today, we have not yet proof for the existence of any of the HIV proteins. And if you don't have proof for the existence of other proteins, you cannot have proof for the existence of the virus.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus,
How come you guys didn't just show pictures from the gradient?
We first show it from the culture. Just by centrifugation, but not sucrose gradient.
From the pictures alone, though, in culture, you can't prove what virus you found yet.
Of course, electron microscopy is not sufficient to prove you have a retrovirus, that's clear. You need we didn't describe the electron microscopy as a proof. We described the reverse transcriptase activity as a proof it was a retrovirus.
No, reverse transcription is actually very widespread. Something like 50% of the DNA in our cells comes about by reverse transcription. When I said 50%, I'm saying 50% of the DNA came about by reverse transcription, but it's not all retroviruses.
The problem is that they detected non-specific phenomena. Reverse transcription, particles, and we've seen all the problems with particles, and antigen-antibody reactions. And so you can't take a whole lot of things that might be something and turn them into that something. That's the problem. If you're walking down if you walk into an empty space and you find an engine block and a fan belt and a generator lying on the ground, what do you say you've got? Do you know what you've got? I mean, is that a car? Could it be a boat? Could it be a plane? Could it be something someone uses on a farm to lift grain up? It could be all those things. You can't make something specific out of a whole lot of things that are non-specific. So they used a whole lot of non-specific and just preferred to believe that this is what they'd found.
What Montagnier did is similar to a fisherman throwing his net into the ocean, pulling it back, looks at it, finds no fish, and yet he claims that not only that he has fish, but he has nothing else but fish. And all of the same kind, HIV.